Hi everyone, my name is Alicia Vanstone and I have done my project on identifying the amino acid frequency in COVID spike protein. Now, amino acids can tell us a great deal of information about the polarity, the water affinity of different amino acids, and from the information about the types of amino acids that are contained in a protein, we can gather some additional information about the types of proteins involved. Now, my raw data consisted of the amino acid counts within different COVID proteins. Now, there are 20 amino acids within these counts, and the COVID proteins that my raw data contained was ORF1A, ORF1B, spike, ORF3A, envelope, matrix, ORF6, ORF7A, ORF8, nucleocapsid, and ORF10. Additionally, those counts of amino acid per protein were also included. Now, I want to hone in on spike. Spike is a surface protein on COVID virus particles, and it's a common source for vaccine design. So what I needed to do is to select out the number of counts of amino acids within the spike protein and its corresponding amino acid name to be able to create a table that I could work with to create further demonstrations and graphs. Now, the first thing that I wanted to do was to create a geome call to see how the variety of proteins have different observed occurrences. So, for example, methionine has a very low number of occurrences, while leucine has a very high number. So this helps visualize the spread of the data and how different the specific fingerprint of this protein looks. Now, here we have the same data in a dot plot format, specifically geome jitter. So this expresses the exact same information, but here it's providing a point rather than a bar. So it could make it a lot easier to compare this specific amino acid counts within the spike protein to possibly other proteins. Now, in order to compare this data to different proteins, we need to be able to utilize the frequency instead of the count. So say that you have a really big protein and it has many amino acids. Well, if it has a count of 100 of a specific protein like alanine, then that might look really big compared to a protein that's only 20 amino acids big. However, it contains a really small number of alanine, but it's a greater amount for the percentage of total available amino acids in the protein. So additionally, what we want to do is display this data on top of one another. So being able to overlay different proteins to see how they might compare and contrast from one another based off of the frequency. So here we have the frequency of amino acids within the spike protein, and this is done visualizing using a geome bar. Now, as you can see here, there are many amino acids, so having a key with so many colors, it can be difficult to actually compare the actual figure with the key. And additionally, it's difficult to show another different protein next to it and see exactly the different sizes of the frequency in amino acids. So in order to confront this challenge, I utilized a dot plot, which I'll be showing on the next slide. So here's another geome jitter, and this one looks a lot similar to the previous one. However, it is done off of a frequency. Now here, it's possible to actually jitter the points and add other points on top of it for another specific protein. So if we did ORF1A, it would be a lot easier to visualize how much higher, say, leucine count or frequency is compared to the leucine frequency within the ORF1A gene. In conclusion, the geome jitter might be able to show the frequency in comparison of different proteins more easily and would be the best choice for this project.